All right, okay. So hello dears. Hello, yes. Good day, yes. Kamusta kayo? Yes, I hope you're all doing fine. All right, and welcome to another pre-recorded lecture in our class in Immunology and Serology laboratory, no? And this lecture, what we're going to talk about is another test to determine or to test our innate immunity. Yes, our very hot immunity, innate, charot. Okay, Mark, wow. Fail. Okay, anyway, innate immunity, yes, na mga tests. The first one, di ba, lumra siya na lecture, yes. Para po, no, dili ko sige pakitag face niyo para po ma-miss ko ninyo. Charat, char, assuming. Anyway, yes, the CRP, yes, the C-reactive protein, yes, that's one of the components of your innate immunity, yes, di ba? Its function is similar to an antibody, but it's not necessarily an antibody. It's just a protein, okay? And again, it's the most widely used, most widely monitored, okay? Acute phase reactant for inflammation, diba? CRP, because it increases about 1,000 times the concentration. So usually, if increase mo CRP, diba, it means that you have an inflammation ongoing inside your body, all right? And for the second one, for second lecture, this lecture, what we're going to talk about now is the complement system, okay? Not all of it, no, but mostly on how to detect or the test that we perform in the laboratory for the complement, okay? So what is again complement system? What are the complements, no? And again, this is still part of your innate immunity. When you say again innate immunity, it's the functions or the effects of the complement system are non-specific, no? Wala sila ginapili, wala memory, all right? And it's standardized. It's the same for all pathogens or antigens, okay? So again, this is the complement system. Back in my third year, this is one of the topics na medyo complicated to understand, all right? But it's carry lang naman, kaya lang. All right, and you'll have more of that in your lecture, the in-depth good na discussion with the, all right? Chudora Cobello, oh, diba? So, carry lang yan. All right, so again, this is complement fixation. Okay, all right, now we'll start first with what is complement, diba? So, saan ang complement, sir? Kanang musay kag mga good things about someone, compliment, joke. Dili. All right. So, complement, these are a series, no? 1890s, in the 1890s, this was first discovered by Paul Ehrlich, di ba? Medyo advanced lagi ko, Mark. Yes, wala ka nag-rehearse ay mong presentation. Yes, it's recognized as a heat labile. So when you say heat labile, it's destroyed by heat. It's sensitive by heat. Present again in the normal non-immune serum. So even if walay nahitabo na infection or something, it's normally present in your serum. All right? But again, it's heat labile. And again, ayan na, si Paul Ehrlich, coined by Paul Ehrlich as such because it complements the activity of antibody in destroying microorganisms. So yan, naka-discover siya ani na proteins, no? Same, it amplifies or it copies in a way, or again, it complements the activity of antibodies against the destroyants against destroying microorganisms. All right, so that is why, hence, no, Paul Ehrlich coined the term complements. But only in 1919 that Jules Bordet, all right, Jules Bordet, he was awarded for the Nobel Prize for elucidating, no, the effects of complement. So intensive study of the complement, no, and all that. So he won a Nobel Prize for medicine. Yeah. Jules Bordet. Ayan. So as you can see, diba, if kabantay mo, usually ang atong mga start yun sa atong presentation, it goes back to history, no? So kinsa itong mga people behind it, no? What did they do? Unsa ilang contribution to science in a way? Because again, as mentioned ako, Jude, as I always mention, history yun is important in ISBB. I mean, I'm not saying that it's not important for the rest of the fields of medical technology, but usually in the board exams, the questions on history, no? It comes or it appears in IS, no, Immunosero or NBB, or in microbio. All right, but mostly good ISBB. So because again, no, we ought to thank these people. Okay, no, they have the brain cells. They had the brain cells, no, and yeah, the effort to really study this, no. And karon kita nag-suffer. Charot lang. Okay, ato na siya gitunan, joke. Yes, thank you again, Jules Bordet and Paul Ehrlich. All right, so that's just a brief history. All right. Ayan. Now, what are again complements? This is a series of proteins, actually 30 ka-proteins. Ayan. 30 ka-proteins that are activated, no? And again, normally present and whose overall function is mediation of inflammation pa rin. All right. So, appeal dyan po siya sa inflammation. Complement proteins, the major humoral, when you say humoral, meaning it refers to fluid, component of your natural or innate immunity, di ba? Back in our previous lecture on antigens, antibodies, we have defined the two types of immunity. You have the natural and adaptive, all right? And each of that nasa like subgroups na cellular o humoral. So when you say humoral, it's more on the fluid part, so mga soluble substances, all right? And for the natural immunity, complements ang major component of, major humoral component of the natural immunity, all right? Okay. And its major functions, number one is of course opsonization, next is chemotaxis, and the ultimate goal, good niya, no? The ultimate end goal or the end event, no, the main event, yes, of your complement system is cell lysis. All right. So, overall, these three functions, ang ilang yung goal is the clearance of your antigens, no. Patsun yun nila. Opsonization, by the name itself, i-cover niya ang antigen by the complement proteins para mas notice siya by the phagocytes and all of your other cells in the natural immunity. Chemotaxis, by the name itself, chemotaxis, it releases signals, no, in a way, so that the phagocytes rather can go to the area of inflammation or infection. And of course, cell lysis, by the name itself, butasan niya, yung buslutan ang cell or pathogens, of course, promotes the influx of water and all that, so ma-imbalance na ang environment inside of the pathogen. Hence, it will lead to the bursting or the lysis of the cell. All right? Ayan. So aside from this, no, na clearance of foreign pathogens, it also, the complement system also works as a housekeeping role. Ayan. So, what do you mean by housekeeping role, sir? So, if na yung mga cellular debris, no, kana mga immune complexes, ayan, na nahitabo or na-form sa imong body, siya po ang in a way mo clear anak, alright, para mawala siya sa body, alright? So, na siya housekeeping roles, naapod siya kaning antigenic or like anti-pathogenic, sakto ba, na effect or na role, okay? Alright, but again, the complement system, because of its effects, no, because again, it mediates, no, or yung ipalabi, or yung it's pro-inflammatory, okay? So, na inflammation mahitabo. That is why there is also regulation of your complement proteins, because if pasagdaan siya, it will also lead to adverse effects in your body. Kay pronounced inflammation, dugay na inflammation. So that will be detrimental, no, and harmful to your body. All right. So of course, in your lecture, you will know the different proteins then found in the complement in your body that serve as the regulation or the regulators of your complement system. And also, if there are deficiencies in the complement proteins, in the complement system proteins, the patient is also prone to different infections and different mga bacterial infections, mga malignancies, and all that. Because again, your complement system or your complement proteins play an important role in the clearance of your foreign antigens and pathogens. Okay? Alright, so as you can see, no, bahalag complicated siya or bahalag part siya natural immunity, it's still important because too much of it is deadly. Too little of it is also deadly. Ayan. So, di ba? So, ayan, do not go beyond and do not go less than your efforts, guys, ha? Char, yes. Sa inyong love, charot lang. Okay, alright, joke lang. Alright, okay, so again, that's the major functions of your complements. Okay. Ayan. Now, for the complement system, we have three major pathways, alright? The first one is, of course, the classical pathway. No, dili siya classical music, EMS. Classical pathway. And this classical pathway involves 9 proteins, alright? And they are triggered, or they are activated when they see an antibody and antigen combination. So, the usual, no? If you're inside your body, example na mo yung nakita na, let's say, antigen, ayan, and antibody na combination, alright? So, this will then be discovered or ma-trigger ang classical pathway ana niya. So classical, so I'm not sure if unsay meaning sa classical, pero like it's I think the first to be discovered muna siyang classical or siya ang old something, okay? Basta classical pathway yung name. And we have the second one, which is the alternative pathway. So when you say alternative, so siya ang second murag pathway, yes, alternative. This one, it does not necessarily need an antigen-antibody combination. It's only triggered by the different components of your pathogen, bacterial cell wall, yeast, viruses, the virus-infected tumor cells, and some parasites. So again, part of the natural or innate immunity, it cannot be destroyed I joke, pathogens can be destroyed in the absence of the antibody. So, it's not necessary that you need an antigen-antibody combination. So, mag-trigger na yun ang kanisya na pathway, alright, in the presence of bacterial cell walls and all the different components of your pathogens. And the last pathway is the lectin pathway, also known as your lectin pathway. Ang alternative di ay, guys, nalimot ko sa other name niya, is also known as the properdin pathway. Ayan, properdin, which is an important protein inside of it. Before Manggol, they thought that the properdin is the activator or dito ma-activate ang pathway in the presence of properdin. But later on, they discovered that properdin is only part of the pathway. It's not the triggering part. Okay? Alright, ayan. So again, sa lecture niyo, i-discuss og todo, guys. Alright, ayan. Ipapaubaya. Yes. Josh Leah, R.I.P. Charot. Ipapaubaya ko na kay Ma'am Teddy, yes. Yan na ng prowess. Okay. Alright. And the last pathway is, of course, the lectin. And according to Stevens, it's also considered to be maybe the most ancient daw, yes, lectin pathway. Maybe it's not the first that was discovered, but it was the oldest yun na nahitabo na pathway inside of our body because it's triggered by, again, mannose or related sugars. So mga sugars na asa microbial cells. And we have what we call a mannan or mannose binding lectin, MBL. When you say lectin, these are proteins that bind to carbohydrates. All right, so you have MBL, mannan binding lectin, mannose binding lectin inside our bodies that bind to the mannose or related sugars in your microbial cell walls, all right? And once this MBL bind to this mannose or related sugars in the microbial cell walls, then the pathway, lectin pathway is then triggered, all right? Okay, actually the lectin pathway is very simple lang siya. Dili kay siya daghang components, if I'm not mistaken, if wala ko naka-forget, no? Out of the three pathways, ang alternative ang medyo confusing for me, all right? And we're not focusing that here. Ang ato focus is the classical. Ang classical is carry lang ang classical pathway. But ang pinaka-simple, if I'm not mistaken, is the lectin. Gamay ra kayo ang components, all right, the necessary for it to padulong sa cell lysis. Because again, all three pathways, although different ang components, the end goal gap po nila, guys, is the cell lysis, all right? Lysis of your cells or of the pathogens. Okay. Okay, all right, ayan. All right. Lectin, again, any group of proteins that are not antibodies but they bind to carbohydrate receptors. All right. Now, we go into the classical pathway. Ayan, so nasa yung mga different stages, nasa yung mga different levels, may ganung level. All right, okay. You have number one is the recognition unit, which is composed of the protein C1. All right. Recognition unit meaning the unit that, this is a protein that recognizes, all right, or that notices the pathogen, all right, or the antigen. Okay, C1. And the C1, din siya sa Jollibee, guys. Ha? Actually, kanina mga proteins din mo na siya kanina mga combo meal sa Jollibee. So I think ma-amaze mo or maganahan mo. All right. So C1, it's composed of subparts, no, or subsets. It's composed into C1Q, C1R, and C1S. Okay? All right. So C1Q, C1R, C1S. All right. So yung mga sub-proteins pa dyan or sub-components. Okay? That's the recognition unit. And this is the recognition unit. So actually, di ba, mura siya o antibody, but it's not an antibody. Okay? All right. Iyahang structure, di ba? All right. Next, of course, once ma-recognize na siya, of course, ma-activate na. So, na-unit na mo activate. So, murag, siya na mo padayon sa cascade, in a way. Cascade. So, when you say cascade, magpadayon siya ba? Like, from one protein to the next, to the next, to the next. Mura siya cascade, diba? Alright. And so, activation unit, so it starts with C4, C2, C3. Alright. Diba? So, parang siya mga combo meal sa Jollibee. Okay. Alright. And lastly, we have the membrane attack complex, which is the MAC. Oh, yes, nanapod si MAC. Mga past natin, guys, it's haunting me. Charot. Ang dami niya dito sa medtech, di ba? MAC sa Bactem, Maconki. You have also MAC here in the complement system, yes. The membrane attack complex. By the name itself, membrane attack complex. It's the complex that forms na on the membrane of on the cell membrane or on the outside of your pathogen, which then leads to the formation of a pore. When you say pore. Dili a pore, ha, Char? A pore. So, murag buslot. So, kani siya ang complex na ang buslot sa membrane. alright, of your pathogen, which then leads to cell lysis. And the remaining proteins are your C5, C6, C7, C8 to C9, okay? Alright, ayan. So, each protein, guys, okay, alright, okay, tama. So, these proteins, all of these proteins, guys, are all synthesized by, of course, the liver, hepatocytes, because again, these are proteins. But except for C1, which is synthesized by your intestinal epithelial cells, ang C1 lang, okay? C1, intestinal epithelial cells ang nag-synthesize. But the rest, they are synthesized by your hepatocytes or sa liver. Okay, alright, ayan. So, okay. The remaining proteins after C1, guys, alright, especially the 4, 2, 3, and 5, they have also sub-components or subgroups, pares ni C1. But for C4, C2, C3, and C5, their subsets are known, their subsets can be C4A or C4B. So, parehas na siya sa C2, C3, and C5. They have also C2A, C2B, C3A, C3B, C5A, C5B. Ayan. Now, usually, the letter A, guys, usually ang A ang naay enzymatic activity, alright? And usually siya pud ang bigger, alright? Yeah, siya ang tama siya. Ang A ang naay wait lang. Yes, okay, alright. So C4A and C4 B, mali, okay. Sorry, erase. Okay. Usually there ang B ang naay enzymatic activity and siya pud ang bigger. Ang letter B ha, ang letter B. And usually, you'll know later sa pag-activate sa classical pathway, the A usually goes out into the serum, alright? Or goes out of the system of the pathway. Except lang for C2, which is ang ang C2A ang enzymatic, and siya po ang magpabilin, kaya siya ang heavier, alright? But the rest of the proteins, usually, ang big yun ang magpabilin, okay? Because again, siya ang bigger, and siya po ang enzymatic activity, okay? Alright, please take note, okay? Alright, sorry, erase yung kanina, okay? Ang B di ay ang enzymatic activity, siya po ang heavier, alright? Okay, example lang for C2, because ang C2A, ang A ang naay activity, and siya po ang bigger, alright? There have been studies na i-change ang nomenclature or naming, but I don't know, wala pa siya na-push through siguro. Alright. So again, that's the classical pathway and the different proteins. Okay. And this is the membrane attack complex. As you can see, multiple copies. Ang C9 ang daghan. Alright. And then ang C8 pod, you have alpha, beta, and gamma. And you have ang C6 and C7, usually usara siya ka-protein. And then C5b. Yes, you'll know later how the classical pathway works. Okay. Alright. Ayan. Okay, now we go now to how it works, no? Or the initiation steps muna. So first is, of course, IgM. As mentioned na, di ba, sa ito ang initial na mga lectures, especially on antigens and antibodies. Your IgM is really the best in complement fixation. Why? Because IgM requires only one molecule because it's a big antibody, di ba? Pentamer. So daghan siyang scope, daghan siyang FC fragment, yes. So, daghang pwedeng complements na makatapot, di ba? So, only one molecule. Whereas for IgG, it requires two molecules. So, best due in complement fixation is IgM and IgG. Usually, the IgM and IgG ang involved in complement fixation. The rest of the antibodies, wala na. Alright, mga chika na lang to sila. Okay, alright. And again, the recognition unit, alright, katong C1 na protein, it attaches to the Fc, okay? Fc portion of the antibody. So, di ba, example, I think na picture later. Sige, later na lang. Alright. Antibodies, according to effectivity in complement fixation, is of course, IgM ang pinakauna, followed by IgG3, di ba? We have 4 subsets or subgroups of IgG, followed by your IgG1, and then lastly, the IgG2. Recall that IgG3, di ba, katong i-include sa description sa antigens antibodies, IgG3 is the largest, okay, sa mga IgG na subgroups, alright? So therefore, ganahan man silang dagko, yes, ganahan silang DAX, na antibodies for complement fixation. Therefore, it's only reasonable that IgG3 ang musunod sa IgM, alright? And kinsa may wala diri, IgG4. IgG4 is not included in your, or dili siya mo take part sa imong complement fixation. Okay? Alright. But diba, unsa tong dili ka mo cross sa placenta na IgG, you have IgG2. Okay. Alright. I hope na remember pa, yes. Okay. Alright. Ayan. Okay, so this is the recognition unit. Ayan. So as you can see, that's for IgG and IgGm. And the color blue one is the C1, which is the recognition unit. So as you can see, asa siya na-bind ang FC portion. FC portion. Okay? Sa FC portion sila mo bind ang C1. Okay. Alright. Ayan. And of course, the membrane attack complex, inano yung appearance. So meron siyang jelly ace actually. Meron siyang jelly. So I'm not sure if it's the real thing, no? In real setting, or inano rin ang drawing dire, but it's so cute, diba? Parang siyang jelly ace. Maglami siya kanon. Char. Okay, alright. So hence, it now bores, di ba? Parang it now makes a pore, alright, or a lesion, which results to the influx of water. So, of course, di ba, dili na equal ang environment diri, mahimo na siyang hypotonic. Okay? Because again of daghan na kayong water, di ba? Diri sa sulod. I'm not sure if hypotonic or hypertonic. Wait lang. Sorry, guys. Okay, sabaw. Alright, basta ang point is masulod ang water. Alright, so uneven na or unequal ang environment sa sulod, which then leads to, of course, ultimately, the cell lysis. Okay. Alright, so again, recall, di ba, IgG, duha jud ka molecule, whereas ang IgM, it's really usa lang yun. So, ato na kita sa gamay. Na diri na part ha, diri lang na aspect chart. Ato ta sa gamay ra na need of antibody molecules to activate, which is IgM. Okay, the best. All right, ayan. So for the next video, we'll now look into how the complement, the classical pathway lang ha, works, and then finally the testing of the complement fixation. All right? Okay, ayan. So I'll see you on the next part. I'll see you on the next video. Okay.